The primary goal of every cell is to stay alive. We're all about efficiency. So if we look at the metabolic pathways in our cells, you'll find that they're very frugal in nature. The cell tries to be as efficient as possible and conserve as much energy as possible. Energy, generally speaking, is going to enter the cell in terms of a carbon-hydrogen bond. So careful management of these carbon-containing compounds is important for the cell to be efficient and competitive in a very cutthroat natural world. The catabolic pathways, so the pathways that break down big molecules into smaller molecules, are going to contain intermediate compounds or metabolites that we can then funnel sideways to alternative chemical pathways to make larger chemical compounds through anabolic pathways. So if we start off with one specific glucose molecule, it'll generally speaking get funneled through glycolysis and then the Krebs cycle and then to the electron transport chain at the very end. But there's individual or intermediate chemical species, and those intermediate chemicals or molecules can be used for intermediate or side chemical pathways or metabolic pathways. This phenomenon of pairing catabolic with anabolic pathways and being able to switch back and forth between catabolism and anabolism is known as amphibolism. Amphi being a prefix meaning either or. So we can start off with our typical glucose molecule, getting funneled to glycolysis, then the Krebs cycle, and then the electron transport chain at the very end. So if we head down, it's in catabolic pathway. But there are some chemicals that are made during the Krebs cycle, or from pyruvic acid or from acetyl-CoA, that get funneled up towards some anabolic pathways. We can use some of these intermediate chemical compounds to make proteins, or other lipids or fats, or nucleic acids, or some polysaccharides. To make our macromolecules, we have two ultimate sources of the monosaccharides that we use to make the polysaccharides. Our monosaccharides, amino acids, fatty acids, or nitrogen bases, so the monomers, the individual building blocks that we make our polymers out of, can either come from outside of the cell as something that was consumed, or the cell can synthesize the individual monomers that it needs through individual anabolic pathways to make larger macromolecules. So we could either eat the individual amino acids that we need, or the cell can make the individual amino acids that it needs to make larger protein molecules. Some organisms are going to be able to synthesize their own building blocks. If an organism is able to synthesize its own monomers, its own subunits or building blocks, it needs to have the correct genetics to do so. Some organisms do have the correct genetics to make all of the building blocks they need. Other organisms do not have the correct genetics. We as human beings are able to synthesize most of our subunits, but not all of our subunits. And then if we look at bacterial species, they are typically able to synthesize all of the subunits that they need to make. So let's talk about carbohydrate biosynthesis. Glucose is a central figure, a key figure in this metabolic pathway. It is going to get funneled through glycolysis, then to the Krebs cycle. And there are going to be many chemical intermediates through this process. We'll use glucose either directly to make cell walls or other polysaccharides. If we could take that intermediate compound from glycolysis, glucose 6-phosphate, which is the second chemical generated in glycolysis, glucose 6-phosphate is used directly by animals to make glycogen, which is in human beings how we store carbohydrates in our liver. If we take fructose 6-phosphate from the glycolysis pathway, we can use fructose 6-phosphate to make peptidoglycan, which forms the cell walls of bacteria. Ribose and deoxyribose are two carbohydrates that are made from glycolysis metabolic intermediates, and then the polysaccharides that make up the glycocalyx of bacterial cells, and the capsules of bacterial cells are made of repeating glucose monomers. So glucose is used for a lot more than just making ATP. We can make many structural molecules from glucose. And the same is true for proteins. Proteins make up a large proportion of the cells. For humans, proteins make up over half the dry mass of the cells. For bacteria, proteins make up approximately half of the dry mass of the cell. Proteins are the main component of an enzyme. Enzymes are defined as a protein that acts like a catalyst. Proteins are also present in the cell membrane, cell wall, and cell appendages. All proteins are going to be made of 20 individual amino acids. Some organisms, like E. coli or most bacterial species, are capable of synthesizing all of the 20 amino acids that they need. So these organisms, or these, generally speaking, bacterial species, make all of the amino acids that they need. We as human beings and most other animals are going to be unable to synthesize some of the amino acids we need. These amino acids that we as human beings cannot synthesize on our own are known as essential amino acids. And these are amino acids that we can only get from dietary sources because we cannot make them in our cells. We also have nucleic acids. Those nucleic acids which are made from repeating nucleotide subunits. DNA and RNA are going to be responsible for the transmission and storage of genetic information or hereditary information through the cells. This hereditary information directs protein synthesis directly. As we put in our cells together, you can think of the parts of a bacterial cell as being synthesized continually. There's constantly new product being made on the production line or the assembly line. Catabolism is also taking place at the same time. So the bacterial cells, or the cell, is constantly breaking down big molecules into smaller molecules and constantly taking those smaller molecules and building them up into new, bigger molecules. Cell division will occur once there's been enough anabolic reactions to build enough macromolecules to serve two cells. So we'll have cellular division occur when the cell has enough building blocks in it for two separate smaller cells. Another key prerequisite before cellular division occurs is we need to duplicate the genome. We need to have DNA replication occur. And then finally, we need to increase the cell membrane and the cell wall. So there needs to be a lot of anabolism taking place. We need to have a lot of manufacturing. The catabolic processes provide all the energy needed for the anabolic processes. The cell gets all of its cellular energy from catabolic reactions by breaking down larger molecules. So, concept check class. If we look at a cell that can integrate anabolic and catabolic pathways, so molecule using and molecule building pathways, to improve efficiency, what is that known as? Is it known as anabolism, amphibolism, catabolism, metabolism, or none of the choices is correct? You can rewind, or you can flip back in your PowerPoint, or check your textbook for the answer. 1, 2, 3, 4, 5. The best answer is amphibolism. Amphibolism specifically refers to combining the combination of catabolic and anabolic pathways. A technically correct answer is metabolism, but that's not the best answer. Metabolism is an umbrella term that refers to any and all chemical reactions in the cell. But the best answer for this specific question is amphibolism. Amphibolism is the pairing of anabolic with catabolic reactions to improve efficiency in the cell. If you have any questions, please post them on the discussion board or shoot me an email. Happy studies!